Hello, and welcome to the JASN podcast, the monthly podcast where we highlight an exciting paper from the current issue of the journal. These papers may be clinical, basic, or translational. I'm Dr. David Ellison, Professor of Medicine and Chemical Physiology and Biochemistry at Oregon Health and Science University in Portland, Oregon, and a deputy editor of the journal. Today, I'm joined by Dr. Christiana Adebayo from the Laboratory for Pediatric Nephrology at the University of Leuven in Belgium. She's the first author of an interesting paper that is appearing this month in the journal entitled Expression of Apolipoprotein L1 Risk Variants at the Plasma Membrane and Haplotype-Dependent Cytotoxicity. Dr. Adebayo, please introduce yourself and tell us a little bit about your background.
Hi, Prof. And everyone. Thank you so much for this opportunity to be here today. I am Oyindamola Christiana Adebayo, a postdoctoral researcher at KU Leuven, Belgium, working on sickle cell nephropathy. I became interested in this field growing up in Nigeria, a country with the highest prevalence of sickle cell disease in the world, and seeing how this disease results in various multi-organ dysfunction, including kidney damage. Very little is known about the mechanism by which sickle cell disease lead to different complications by which sickle cell disease lead to kidney dysfunction. However, what we know right now is that when this patient develop kidney damage, they are less likely to be given a kidney transplant, which warrants research to understand what is going on in this population. What even makes my project more exciting is also the fact that West Africa, particularly Nigeria and Ghana, has the highest prevalence of Apolipoprotein L1 Risk Variants. So over the last decade, I've been very fascinated by this intersection, and I've been very fortunate to have been mentored by exceptional interdisciplinary experts in the field of hematology, nephrology, and ion channel research by the names of Professor Elena Levchenko, Professor Vela Bach, Professor Joris Vins, and Professor Labatus van der Hoeven.
Great. Well, we're delighted to have you here. So I'll start out with the first question. I think it's likely that all nephrologists are aware of the importance of the risk variants in APOL1 by this time and their role in glomerular sclerosis and chronic kidney disease. But some listeners may not know much more about the variants in terms of what they are and what populations they're found. You mentioned that already, and their beneficial and detrimental effects on podocyte function. So if you could talk a little bit about what does APOL1 do normally, and what are the proposed mechanisms for toxicity of the variants? I know there's still a lot of debate about whether they're gain or loss of function variants. Perhaps you could provide some background about this briefly at the beginning.
APOL1 plays an important role in innate immunity. It is part of the high-density lipoprotein, which circulates in the body and protect humans against trypanosome, which is transmitted by sesafly and causes sleeping sickness. However, two subspecies of trypanosome, which is the gambiense and rhodesiense, have found a way to get around this protection. And in response, the human gene has also evolved through positive selection, and this has resulted in two variants, the G1 and the G2. The G1 variant is as a result of two nucleotide substitution, and the G2 is as a result of two amino acid deletion. When we look at the prevalence of these risk variants, the G1 and G2, they are mostly found in African population, specifically the people of West Africa. And this is because in this population, the two subspecies, the gambiense and rhodesiense, are also very endemic. But what we know generally from the case of like sickle cell disease, whereby hemoglobin has protect against malaria or result in a very dangerous and genetic disorder, is the same that happens now in the APOL1 risk variants, whereby when you have two copies of these risk variants, you have an increased risk of having kidney disease, such as the HIV-associated nephropathy, hypertension-attributed nephropathy, and also COVID-associated nephropathy, FSGS, and even sickle cell nephropathy. And when we look at the mechanism, we have to be conscious that not everybody that has the APOL1 Irish genotype develop kidney disease. And also, it has been shown that does APOL1 has a function in the kidney or does not have a function, leading to whether APOL1 is a gain of function mutation or a loss of function mutation. But right now, most of the evidences has been going towards the gain of function mutation. And this is because we are, our closest relative, which is the gene, does not have an APOL1 gene, suggesting that probably there is no protective effect there is no role outside of the protection against trypanosoma it offers. There is no function of APOL1 within the kidney, since higher primates, it's only few higher primates that expresses it. Another evidence is also from India, whereby they saw that, they saw in a person with a non-mutation in APOL1, which causes the loss of APOL1 gene itself, having a normal kidney function. So with these two evidences, it has really made researchers to think is a gain of function mutation. And also when we look experimentally, when you overexpress APOL1, particularly the risk variants, you see an increase in cytotoxicity, which have been mediated via the increase in poor forming activity, also by dysfunction in the mitochondria, also inactivation of APO3, and as well as activation of inflammasome, for instance. So all this evidences suggest that APOL1 is a gain of function rather than a loss of function mutation.
Great, thank you very much. That's very interesting. One other aspect of the background, a bit about which I was unaware, was the impact of the sort of background that the APOL1 variants exist in on pathogenicity. So you state that APOL1 risk variants expressed in the native African haplotype exhibit greater cytotoxicity than those in the reference or wild type backgrounds. And so it sounds like there's no one wild type background for all APOL1. It varies from population to population, and that it may be more the variants may be more pathogenic in some backgrounds. Could you discuss that a little bit, please?
Yes, you are right. I will actually start by defining what a haplotype means. A haplotype is a series of genetic variants that are inherited together as a block. The human APOL1 has at least 7 common non-risk haplotype with different coding sequences. So there is no canonical wild type APOL1, but rather many non-risk haplotype, which differs from one another by at least one amino acid to several amino acids. In our present study, we majorly focus on 3 of the haplotype, and those are the widely studied ones, which is the African, the reference haplotype, and what we call the wild type. But now we have changed it to be the worldwide common haplotype.
That makes it much clearer, because it was said the reference haplotype, and that was a little unclear to me why it was the reference.
Yes, so now it's the worldwide common, and it's present in approximately 65% of the population. So while the others are present in, it's more or less frequent. So, but now we already changed it to worldwide common during the production.
Great.
Thank you for your input about that.
So it sounds like the control APOL1, when you expressed it in cells, you observed it in the endoplasmic reticulum, but also at the plasma membrane. And then you used recovery after photobleaching. And it sounds like that compared with the wild type allele, the G1 and G2 did not recover as completely after photobleaching. So maybe you could discuss those findings and what you think they mean.
In this particular part of the study, we wanted to show 2 things. The first thing is the localization of APOL1 variants, and the other thing was the trafficking of APOL1 variants. So for the localization, we showed in our study that APOL1 variants are localized at the plasma membrane and also in the endoplasmic reticulum, using fluorescent staining, immunofluorescent staining, and plasma membrane isolation technique. But at the moment, the presence of APOL1 at the plasma membrane still remains like conflicting. And we wanted to understand if indeed APOL1 is present at the plasma membrane, and if it's present, the traffic into the plasma membrane. What is the mechanism by which traffic? And to do this, we use a very interesting imaging technique, which is the total internal reflection fluorescent recovery after photobleaching. With this technique, you can really visualize something between the 100 to 200 nanometer thick, which is around what you expect near the plasma membrane. And with this technique, we are able to show that APOL1 are actually present at the plasma membrane, and the risk variants, the G1 and G2, they have a large, less mobile-like and vesicle-like structures. And also, they have reduction in recovery when we photobleach, which suggests that probably there is something going on with these two variants, the risk variant, the G1 and G2. Probably their traffic into the plasma membrane is perturbed. And in order to even further to look into the molecular mechanism of the trafficking, we also introduced the N264K mutation, which is the genetic modifier of APOL1. And with this mutation, we saw a restoration of the trafficking thing, because we saw an increase in recovery after photobleaching and also mobile vesicles in the G2 transfected cells containing the mutation. And another thing we also went ahead to do was to further probe into the trafficking by treating the cells with cytochalasin D and actin filament, cytochalasin D to destroy the actin filament, and locodazole to destroy the mitoprovolins. And again, we saw, especially in the G0, that upon treatment of the cells with cytochalasin D, and then performing a photobleaching experiment, the FRAP experiment, there was a reduction in recovery after photobleaching, suggesting that the trafficking of APOL1 to the plasma membrane might be true. It's mostly through the actin filaments. And in the G1 and G2, we did not see a statistical significant reduction after treatment, but this suggests that probably the actin filaments in this G1 and G2 already been disrupted, which is something that is currently known in literature, that actin cytoskeleton is also affected in high-risk genotype podocyte, APOL1 podocyte. But I have to mention right now that why we show this, the whole mechanism driving APOL1 mediated APOL1 risk variants to the plasma membrane is incompletely understood, and there is more work that needs to be done in this regard, especially with the characterization of the vesicles. We really have to know what are they. Probably they will also share more light into the trafficking of APOL1 to the plasma membrane.
Great. So move on to a little bit more of the results. On figure 3, you were showing differences in cell calcium concentration. I guess the understanding is that the APOL1 can act as an ion channel and allow calcium to come into cells. And when you transfected with G0, calcium levels went up, but that was higher in what you call an EIK background. And this difference was magnified when the risk variant was expressed. So just again, because I'm naive about this, the EIK background is common in people of West African heritage versus other backgrounds. Is that correct? And could you discuss the importance of this finding?
Yes, the EIK haplotype is the African haplotype. The EIK is just the amino acid. It's denoted by the amino acid, like glutamic acid, which is at the position 150, isoleucine at position 228, and lysine at position 255. So all the names are based on the amino acid substitutions in the APOL1 gene. The African haplotype is mostly common in people of African heritage, while to a lesser extent also in the mixed American, Latin American population. And the most relevant part of this particular part of our work is the fact that we can now see that why the risk variants are really important for mediating kidney. We should also consider the haplotype they sit on, because even G0, like we show in our paper, can be toxic when expressed in a different haplotype, like the African haplotype. And also, when you look at the African haplotype compared to like the worldwide common haplotype structurally, the only difference between both haplotype is just an amino acid at position 150. In the African haplotype, we have a glutamic acid at position 150, while in the worldwide common haplotype, we have lysine, which is a positively charged amino acid at position 150. So the difference in charges might lead to structural conformation in the protein, which has increased the pore forming activity, which we measure through increasing intracellular calcium concentration, which was driven from influx of calcium from extracellular medium.
Yeah, I think this is really interesting that there may be an interaction between the APOL1 variant and the haplotype it sits on that's much more complex than I had previously thought it was. So let's move on a little bit to therapy. So efforts to find therapeutic approaches to APOL1 mediated kidney disease have been ongoing since the variants were discovered, but there's recently been a lot of excitement surrounding cell, animal, and human phase 2 studies of inaxaplin, which is, I guess, VX-147, which is a specific APOL1 inhibitor. And there was a high impact paper in 2023 in the New England Journal of Medicine. So the drug inhibits APOL1 channel function, which was, as we've just discussed, proposed as one of its mechanisms of action. Given that success in those studies, albeit there's not been proof that it improves hard outcomes or reduces progressive kidney disease, what were the unanswered questions regarding APOL1 that you wanted to answer using the cell culture model? And I would just say that it seemed from the paper like the drug did reduce calcium uptake by cells, which is what you'd expect if it's a gain of function mutation and the drug is therapeutically beneficial. So maybe you could talk a little bit about what you were looking for in terms of the drug studies.
A major question we aim to address using inaceplin was to further elucidate the source of calcium. At the moment, in the field of APOL1, this is currently debated, with some groups suggesting extracellular calcium plays an important role in cytotoxicity, while others believe that calcium is coming directly from the endoplasmic reticulum. In order to address this issue, we use various approaches, including pharmacologic inhibition with inaceplin, to pin down the source of increasing cytosolic calcium. Using single-cell calcium fluorometric analysis, we are able to observe in our transfected cells at real time, upon perfusion with 1 millimolar inaceplin for 3 minutes, an instant drop in calcium level. And this, for us, suggests that APOL1 is a non-selective cation pore, and it allows the influx of calcium from extracellular medium, and this blockage can be stopped, and we can really visualize it at real-time level. And with this, among other results, we then propose that actually the channel is not only a monovalent pore, as suggested by Dante et al., but actually a non-selective cation pore that will allow the flow of other cation in and out of the cell. And I believe that this also opens new avenue for further drug screening to further identify other pore blockers that can help to ameliorate the effect of APOL1 overexpression as well.
Great. That's very clear, and that's an exciting finding. So next you confirmed, so the experiments were done up to this time, I guess, in HEK cells, but you confirmed a lot of the results in a podocyte cell line. So I was wondering why you decided to use the HEK cells first and then confirm them in the podocytes. I assume it has to do with the fact that podocytes are probably harder to transfect and harder to use. So what additional information did you gain from repeating some of the experiments in podocytes?
Yeah, you're totally right. We used the two models. Actually, Egg cells are the most commonly used cellular model in APOL1 biology because they are rapid, they are very easy to transfect, they are very easy to maintain, and they proliferate really rapidly. Even though they provide a very valuable platform to elucidate APOL1 mediated kidney disease, they don't endogenously express APOL1, and they also lack specialized structures that mimic what like that of podocytes, which is the main target of APOL1 mediated kidney disease. So we wanted to ensure that we can translate the data we observed from the egg cells directly to patients and be able to mimic the microenvironment of what we see in patients with APOL1 mediated kidney disease. And in order to do this, we use two different podocyte models. We use a stable cell line, which expressed a single copy of APOL1 under the doxycycline-inducible promoter. And also we use the patient-derived podocyte and its isogenic control to further elucidate APOL1-mediated kidney disease upon stimulation with poly-IC. And we believe these two approaches give us something closer to the microenvironment seen in pathologies associated with APOL1-mediated kidney disease. And I have to say that what we observed in egg cells was also translated to what we see in the podocyte. So both were actually relevant model, and we were able to validate all data simultaneously with the two models.
Yeah, obviously that's reassuring to know because anybody who's worked with HEK cells knows they're sort of just bags that express proteins, and so they're not very much like real kidney cells. So I guess the last question I have is, in the discussion, you note 3 key takeaways from this work. So I wonder if you could just briefly discuss what the takeaways were and where this might lead in the future.
The first takeaway from our study is that APOL1 variants are trafficked to the plasma membrane via the actin-dependent transport, where they show distinct dynamic properties. The second takeaway is that APOL1 is functionally expressed at the plasma membrane as a voltage-dependent, non-selective cation permissible core that mediates haplotype-dependent cytotoxicity. When we look at the two takeaways, we know now that APOL1 is present at the plasma membrane, and this opens up a new avenue for therapies that directly blocks the protein at the plasma membrane without disrupting other potential functions of APOL1. That said, there's still need for future work to really understand the trafficking mechanism of APOL1 to the plasma membrane and the biophysical properties of APOL1 itself. And when we look at our last takeaway, which is that APOL1 variants expressed in the African haplotype background promotes increased calcium influx into the cytoplasm, enhancing cytotoxicity, and effect attenuated by the N264K variant. This again reinforces the need for genetic screening and patient-tailored management, because a kidney patient with the G2 Risk variant who also carry the N264K mutation would not respond to the same therapy as somebody with just the G2 Irish genotype. Looking at this from the research standpoint, we also have a new control for experiments now with the N264K variant, because the G0 itself could cause cytotoxicity, which we showed in our project, if it's highly expressed and if it's on the African haplotype. So having the N264K will help researchers reconfirm their data in the same risk variant haplotype. For instance, with the G2, you can always do experiment with the G2 variant with or without the N264K. And also, I believe that this will also accelerate other therapeutic discovery for APOL1, because we can really dig into the molecular mechanism driving the protective effect of the N264K. And we already showed it in our own study the trafficking of the N264K was not impaired. So it also still trafficked to the plasma membrane. It's still localized to the endoplasmic reticulum as well. So there's something going on, but we don't know. And this thing can provide evidence and guidelines to other therapeutic discoveries.
Great. I think this has been super fascinating. I've learned a lot about APOL1 mediated kidney disease from the studies. I hope that you have the opportunity in your career as you go forward to move these observations forward. Everybody would love to have some drugs or medications that can be used to slow the progression. And as you point out, also genetic screening and thinking about the background haplotypes is very interesting. And I think that provides new opportunities for diagnosis and intervention. So thank you very much for sharing this work, and appreciate your taking the time to describe it to us. Thanks very much.
Thank you so much.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.